Thank you everyone for joining our webinar today. I'm sure people will be trickling in. We have about 300 people registered for the webinar, but in the interest of time, start. So I will start with a disclaimer. The opinions expressed in this presentation are the presenters and do not necessarily ISoP and or CDISC. And Fang has from FDA that I thanked. So welcome everyone. Start with, why we do read standards for PopPK datasets. So given that there are audience from, this areas, some of you are very familiar with this and you don't need to know. But to level set everyone's understanding, I want to start by emphasizing these datasets, these PopPK datasets are the foundation for the high quality and impactful pharmacometric analysis. And unfortunately, in creating varying requirements and varying degrees of quality, making it very hard to review internally and externally, and it might affect the quality of the pharmacometric as well. And in addition, it will make it difficult to optimize or automate our workflows. And that is why I've picked few elements to highlight here. Standard will bring a lot of efficiency and quality, minimize variability, because the data set is not either tool dependent or modular dependent or programmer dependent. In fact, it's standardized across, and it streamlines the collaboration both internally and the clear implementation guidelines come up in this ADPPK guidance will act as almost like a training document. We all know how difficult it is to hire programmers that have experience with population PK work. We always end up, training them after hiring, and there is no guidance like we have for the rest of the SDPMI. So this will act as that, and it enables automation, which will touch slides, and it will definitely expedite it. So I'm going to take you through the journey of ADPPK. So we've established the data standard working group under ISoP in 2015 with approximately 30 scientists and programmers. And we've done a thorough survey across the industry to understand how different companies are constructing these data sets, what are the quality procedures, and what are the tools that they're using. And in taking into account all that, we wanted to fill the gaps and develop a standard that works for everyone. And we've accomplished that and published our and made it open for review. The whole pharmacometric community has reviewed and commented, and we were able to publish in 2020 at ACOP under ISoP, the first version of PopPK data stack. The next step afterwards was to make it CDISC compliant, because that's more widely recognized and accepted by different regulatory parties. So we established ADaM ADPPK working group in December 2020. We were approximately 20 people from various backgrounds, and we went through line by line, variable by variable, and made sure everything. We made necessary changes to make standard and with CDISC ADaM. It went through very rigorous review, and we were able to publish our CDISC ADaM PK implementation guide in 2023. So just to level set everyone, for those who are not that familiar with CDISC or ADaM, CDISC stands for Clinical Data Interchange Standards Consortium. It's a not-profit organization that takes a rigorous approach in developing and advancing standards in clinical research and beyond. It brings operational efficiency and is widely accepted by various. And this particular analysis data set, our PopPK analysis data, falls under ADaM model. And I invite Luke, who is my co in ADaM working group, to give some fundamental principles of ADaM before I go into the implementation of ADPPK. So I'll stay tuned now. Luke has 18 years of experience in the industry and extensive experience in PopPK. Yes.
Thank you, Neelima. Real quick, some of you may or may not know, but CDISC is comprised of many groups that provide different functions. Two of note for PK are going to be STTM and ADaM. STTM is going to deal with the source data and it's going to regulate domains such as PC and EX. ADaM's purpose is to regulate or support analysis data sets. Analysis data sets can be used for producing TLGs, performing statistical analysis, or in our case, PK analysis. ADaM does this by providing guidance on dataset creation, as well as the metadata supporting that end. The datasets and metadata associated with ADaM provide many benefits. They allow for traceability, the ability to follow trace a record back to the source, the data that also needs to be compatible with available software tools. The ADaM dataset will also clearly and unambiguously communicate the content of the analysis. A little bit about the classes and subclasses that you're going to see within ADaM. The datasets are defined by their classes and or subclasses. The first and most common of the classes is going to be the subject level dataset referred to as ADSL. This contains most of the general information such as age and general level flags. Next, we have the basic data structure. It's referred to as BDS. This is the common structure used. You might see this used to describe labs or exposure. It has a few PK-related subclasses that were newly introduced, and they closely follow the BDS structure. Those two of note are going to be ADNCA and ADPPK. The remaining two structures are OCCDS and ADaM Other. Analysis data is going to be chosen to fit into one of these classes. One thing of note is that the naming convention. It's important to note that only ADSL has a specific naming convention. All other names are up to the sponsor to determine to name. Okay. So diving down into ADaM BDS, this is the structure that is most applicable for population PK. BDS follows a vertical design like that of PC. The dataset design contains one or more records per subject, per analysis parameter, per analysis time point. The typical analysis dataset used in PK includes a stacking of domains like PC and EX. The BDS structure allows the copying of variables directly from the source data or domains. Any variable in the original domain can be kept in the new dataset. You can take advantage of that. So it's always good for traceability to retain those. Any variable in the original domain can be kept in this new dataset. Other benefits include the robust and flexible support for the performance and the review of statistical analysis. In addition, records in the ADaM BDS dataset can be from an observed domain, derived, or imputed as needed for analysis. Additional variables and observations can be added as needed to support the analysis and to provide traceability. The subclasses, we utilize the subclasses so that we can benefit from the stability and traceability provided with the root structure. A subclass follows the original class mostly, but with a few changes that are needed for the analysis. One benefit of this is the ease of conformance checks. There are commercially available conformance check software packages that can determine if your data contains any issues that may need to be addressed before you submit them. This ensures the integrity of the dataset that is used for analysis and prepared for submission. By fitting into a BDS-like structure, each vendor can easily add the ADPPK subclass to its established submission process by utilizing the checks that are already in place. ADPPK was defined as a subclass of BDS in a similar manner to that of ADNCA. The differences from the original BDS being that the necessary variables and content needed for specific PK analysis are added, and Neelima will go more into that in a little bit. A little bit about the processes we went through. We went through the CDISC review process. That is a fairly extensive process. The life cycle of the project it goes through a lot of reviews, and the group has to go through and answer questions and respond to each review. A little bit about the project. Once it reached CDISC, the document was updated to make it compatible with both CDISC standards and also to remain usable for the PK analysis. This involved looking at the naming conventions and the content required by both end users. Once the document was updated, we started the review process. At each stage in the presentation, a presentation to a specific group was made. Each group would then ask questions or make comments, and these comments and issues were then logged into a system for us to look at and to respond to. They're also available to be viewed by other people within CDISC. The ADPPK subteam then reviewed each issue and discussed how to best respond to them. Each step was documented and ensures that everything is responded to properly and not forgotten. The first review was done by the ADaM team itself, followed by the CDISC group, and then finally the CDISC public along with the FDA was allowed to review and look at the document. And in each step, we would meet again and we would look at what was brought up. We would talk and make sure that it was consistent with what we wanted to go forward. Once all of these were addressed, they reviewed that to make sure that we commented on everything, and then they allowed us to publish. That concludes the review process in a nutshell. Thank you, Neelima.
Thank you, Luke. So now you have some basics and foundation about how ADaM works and CDISC works. So I'm just going to make this bigger. So I'm going to give you a little bit deep dive into the ADPPK standard implementation itself. So I'm going to switch back and forth between the implementation guide, which is a PDF. It looks like this, and it's on CDISC website for download. I'm going to work with ISoP and put a copy here because one needs an account in CDISC to go through to download this. So I'm going to start by introducing that, the standard is intended to provide a template for model-based PopPK analysis. It will be utilized as an input into a software package for PopPK analysis. And the underlying structure is based on some of the original PopPK modeling software. And it also contains both numeric and character results. So, one needs to adapt to an individual program. So I'm going to bring the slide deck back to go over some of the details. I'll start with the name of the data set. So we went with ADPPK, and that's the recommended name for these data sets. But let's say, for example, you're working on a combo project and you have two drugs and you're submitting both population PK data sets in a single filing. You cannot name both as ADPPK. So you can use additional characters as long as you stay within 8 and name them uniquely. And included PopPK specific variables like DV, EBID, and compartments. I'm sure we fought hard to keep these because they're not really CDISC compliant, but because all the pharmacometrics community is used to it for their tools, we were able to keep them. So you'll be happy to see that they're there. And we adopted ADaM naming convention for the rest of the variables. And I'm going to highlight only the portions of it that define the structure, content and traceability of the ADPPK here. Starting with the time variables, we all have been used to naming time relative to first dose and previous dose in various ways. But, we landed on using RLT because that's the suffix used for naming time variables in ADaM. So for actual time relative to previous dose, it's APRLT, and actual time relative to first dose, it's AFRLT. And we would not be using the TM as the suffix because it's reserved for clock times and it will cause errors with conformance rule checks. Another new thing that you will be interested to know is the added variables like param, CD, and eval, which are not part of our normal PopPK data set, so you are not used to seeing these. But if you have used other SDTM or ADaM data sets, you're familiar, with these variables. But they're nothing to be scared of. It's as simple as, param CD is the parameter code. For dosing records, you can populate it as dose, and for PK records, you can populate it as PK, and the structure can be extended to include PD records as well. Param is the text version of the parameter. So for the dosing, you can bring it from treatment group, and for PK, you can carry it forward from the PC. If there are multiple, drugs, we can make param CD, drug 1, drug 2, and similarly. Eval is nothing but it's the result for ADL, for lab data set or for the exposure data set in CDISC, not exposure, sorry, for lab and PC. Eval is nothing but it stores the result. And for us, the equivalent variable is DV, which is dependent variable, which stores a concentration value or the PD value result. And some of the other variables that I would like to highlight that, again, goes back to traceability is the flag variables. The first set, which is FLG-REAS and FLG-REASC, are the numeric and character version of the variables. These are primary. The purpose is to identify the record with data issues. So we keep that record. For example, there was a missed dose time. We may use imputation methods and impute the clock time of the dose and flag that record. But for traceability, we would like to identify that record as an imputed record and flag it with a certain number. And you increment so on for different reasons. And the character version of it simply has the text reason in text, and there should be a unique correspondence between the numeric and character values. And the next set of variables are the exclusion variables. So these are the ones that identify the record for exclusion from analysis, but kept in the data set. So the first set, not necessarily you would exclude an imputed record flag or, some other issue. You may choose to keep. But there will be certain reasons where you want to exclude, like Day 1 pre-dose samples or certain BLQ records or certain other data issues. So we would like to keep them all in the data set and flag them with numbers and include a corresponding variable called EXCLFCOM that will have the text values for that number. So this way the data set is self-documented, transparent, and traceable with its issues. And we have provided a list of standard slides in the implementation guide. Another thing is we define source data usage principles. The rule of thumb is to go to the immediate predecessor. So because in preparing this ADPPK dataset, you would access several SDTM and ADaM datasets. And the recommendation is to first use ADaM datasets because that will have the most updated information with considered to any derivations, but in the absence of that, we could start with SDTM variables. Some of the variables that are already defined in other domains are not repeated in the structure of the PopPK dataset in the implementation guide because it's a repetition. We can simply, pull it from wherever they are in other SDTM ADaM domains. Another variable of interest for traceability is the date and time of a record. So for example, for dosing records, this variable UDTC captures the dose date and time. And for PK record, it captures the PK collection date and time. It's incredibly useful, this particular variable, because if there is an issue with the derived times, you can immediately check if there is something wrong with the dates and times. And it helps with the QC process, and it also helps with merging time-varying covariates. If you don't do now, and let's say you come back to that study 6 months later or 1 year later, and you want to add some time-varying information, it would be hard to add if you don't have this information in your PopPK dataset. So that's why I want to point that out. And we also define the naming conventions for covariates. The cov in the brackets on its own stands for time-varying covariate. but with a suffix of base. BL stands for baseline. So WT on its own is a time-varying weight, and WTBL is a baseline weight. So that's the convention that we are going for all baseline covariates. Similarly, COV-N for numeric version of the categorical covariate. And so for sex and sex N, race and race N, like that. And COV-I is reserved for any covariates with imputed values. Again, if we choose to impute a missing covariate in the data set, we'll leave the original variable as is and create another variable and populate that with whatever the imputed values along with the other non-missed values. That's one way to do. And if you're used to imputing in the model file and not touching the data set and source data, that's fine. And you can do that too. We talked about it in the implementation guide as well. And COV-GR, stands for like any grouping variables. For example, if you want to group ages for pediatric studies, especially, age GR1 can be used for group 1, like 0 to 6 months and so on and so forth. So, these variables will come in handy for those type of situations. And another thing to note is we define whether a numeric version of the variable is primary or the character. So since we have included the pairs for race, sex, and all, both the character and numeric, we've defined the character variable as the primary. So that will not have a suffix, and the numeric one will have an N afterwards. So you will see that. And units for variables will be added in labels except for variables like a value. So for all the variables, you would see the units in labels. Because CDISC requires to include a-val, we had to use a-value for capturing the unit of the DV. So it will have PK units and PD units and so on. And here are, I put these as screenshots from our implementation guide. You will find a lot more equations for covariates. These are some typically used covariates and the formulas for them. And each of these equations comes with a reference that you can take a look at. For whatever reason, for your specific analysis needs or population that you're using, this is not the right equation. You may use a different equation, but to start with, we provided recommendations for typical covariates. Handling of missing data. So we have an extensive section dedicated to handling of missing data in the implementation guide. And specific guidelines were provided for each scenario because this is the most common issue with the PopPK datasets, with metadata related issues, especially for oral dose studies due to compliance and due to patient not coming to clinic every time the dose was taken. For all those reasons, there ends up several scenarios on how to handle missing data. So we have a section that goes over for interval doses, how we handle, for continuous doses, how do we handle. And it touches on missing dose date times, missing dose amount, missing PK sample date time, infusion start and stop times, infusion duration and rate, and etc. And again, the recommendation is to flag all the imputed records. So that was the overview of the data sets, and I would highly recommend that you go through the implementation guide. And I want to talk a little bit about how do we transition from whatever standard we are using in our own companies to ADPPK. And I can speak from my experience at BMS that, this is what we've done, and, hopefully, it might help someone. So we started for all new studies with the CDISC ADPPK template, and we've started implementing it from last year, ever since it was published in October, and it's been working fine. But the only challenge is for ongoing and older projects, on how to phase them out. So the way we've been doing is to do the phase out and make the update at the next time we go and look at that data and we update it for some other purpose. So we would have to take those older data sets and map to new standards, which can be challenging and time consuming, but it's a one-time investment for each project, and, and then you won't have to do it again. And you will gain efficiencies. And I would like to point out a resource that's available in open source. BMS released an open source web app that automates the creation of the CDISC PopPK spec. You can find it on GitHub. It's right here. So I put a screenshot of the app where it populates the whole data structure, including the variables, its attributes, the derivations, and any imputation rules and everything. So it takes away the burden from the pharmacometrician to populate all this information and to create a spec. It brings efficiency, it brings quality, and it already has built-in checks, so you don't have to worry about, okay, am I being compliant with the, CDISC ADPPK? And also it has the ability to search and import specs from other studies and modify for use for new project. And it streamlines the external collaboration and internal as well. So if it helps, check it out. I want to, wrap this up by acknowledging all my team members, both in ISoP working group and CDISC working group. And I had the pleasure of co-leading both, one with Adriana and then other with Luke. So without the help of contributions from all these people, we couldn't have come where we are. And in addition to that, several members from all these companies supported offline as well, because these are just the representatives, but we would all go back and talk to our groups and then come back and give their feedback. So indirectly, many of them have contributed, from all of these companies. So thank you so much. And I'm going to transition over to Fang Li now. We've reserved the best for the last. So Fang is a senior reviewer at FDA. And he's going to give us FDA perspective on population PK standards and how we can adopt them. Thank you. And Fang, I'll advance the slides for you.
Yeah, thank you. Thank you, Neelima, for your kind introduction. Good afternoon, everyone. My name is Fang Li. I'm very pleased to be invited to speak submitting population PK data sets from a regulatory perspective. Before we dive next slide. Before we dive into the content, I have important disclaimer similar to Reinbolt did. Please note the opinions expressed here are totally mine and do not necessarily reflect the official views of the U.S. Food and Drug Administration. Before we talk about population PK data sets, let's take a closer look at Pharmacometrics at FDA. Pharmacometrics division is the largest review division in the Office of Clinical Pharmacology. At this time, we have 31 dedicated review scientists. As of 2023, we have 3 review teams. 5 review teams cover different therapeutic areas, and 1 dedicated team for reviewing PPPK submissions, and we have 1 team responsible for TQT review to ensure cardiac safety. With the advance of QSP models and artificial intelligence in recent years, we have specific review staff to handle these submissions there. We are still growing fast due to increased need. We deliver impact mostly in dose selection, confirmation, and optimization in drug development and submissions. We have impact in advocacy, safety, dose changes, pediatric studies. We have impact in benefit risk analysis, labeling, and DDA studies. Population PK modeling and exposure response analysis are the most important tools we are using on a daily basis. In addition to that, we have developed a range of review tools and templates to increase productivity, review efficiency, and consistency. We also have a knowledge management task that develops disease database and the library. We also have an application like e-safety to make sure safety data can be quickly and thoroughly reviewed. Okay, I want to zoom out a little bit to talk about model-informed drug development at the Office of Clinical Pharmacology. in the early times, from 1990 to 2000, that's the time population PK was introduced to drug development. At that time, population PK was only used to identify covariates that could have a potential for dose adjustment. For example, dose adjustment based on different body weight group, renal impairment, hepatic impairment, or by different race. And the pharmacometrics group was at the time just several reviewers. From year 2000 to 2010, that's a period we call rapid growth. In addition to population PK, dose response relationship and the exposure response relationship were introduced to provide evidence of efficacy and safety. And that was the early times that the PPPK research and application was introduced. From year 2010 to this time, pharmacometrics approach and mainstreams and population PK analysis and the exposure response analysis become routine in our submissions and the review work. The important milestone was the PDUFA 6. MIDD was specifically included as a strategy for population PK analysis. From 2010, population PK and the ER analysis are more widely used. At this time, more than 85% of the submission, BLA or LOA and the supplements include population PK and the ER analysis. Also, population PK moved to dose selection in early phases, such as for those for phase 2 studies. Dose extrapolation in pediatric patients is also important part of pharmacometrics. The office introduced MIDD pilot program. That's a new pathway for approval drug without clinical studies or with limited clinical studies. More QSP models and artificial intelligence submissions were reviewed by our staff. So from here, we can see pharmacometrics is an evolving science, and so is the division. If I introduce this framework, it's just to highlight the importance of population PK. From my viewpoint, population PK is the most important component of model-informed drug development. Without population PK, all of these tasks cannot be possible. Here, I list some scenarios that population PK used in drug development and informed drug use. For example, population PK can be used to identify covariates that might be clinically significant or derive exposure metrics for exposure response analysis. In later phase of clinical studies, when only sparse PK samples will be connected. Based on pharmacometric analysis, alternative dosing scheme in clinical studies can be proposed or studied in PMR or PMC. And those we contribute, population PK contribute significantly in dose selection in pediatric studies. Now we move to submitting clinical data for drug evaluation, where we mention submitting clinical data. Mostly this include clinical and non-clinical data sets, clinical and non-clinical study report, and additional supporting documentation and materials. These materials cover a large majority of materials submitted to regulatory agency and reviewed by many disciplines. Along with these materials, modeling simulation and the data and the results are submitted for review by the Division of Pharmacometrics. At this time, almost all clinical studies receive data in CDISC format, but population PK datasets is not. This slide highlights the locations population PK datasets it can be found. This is ETCD. As I mentioned, more than 85% NDA or BLA submissions include population PK analysis and report. Mostly, this datasets are located in module 5.5.3.3.5, population PK data study report and information. Next. Occasionally, less commonly, they are in module 5, but in other locations, such as patient ID and PKPD study reports and related information. I also found it in reports of analysis of data from more than one study. We sometimes in other locations we have to find it. Again, I emphasize that population PK datasets are not submitted in disk format. That's very different from other clinical datasets from other studies. Here, I list the population PK and the related guidance you should refer to in your submission of population PK datasets. That's including population PK guidance or exposure response guidance for industry and the PPPK guidance. In each of these guidance, we details requirements for format and content for submitting. In addition to that, we have a website that updated the latest requirements about submission population PK report or format requirements. You can check that regularly. Here, these slides, I describe how modeling and simulation results are prepared, submitted, and reassembled from the perspective of industry and analysts at the FDA. For pharmacometricians working in pharmaceutical industry, they assemble data, population PK datasets, and develop a model and create figures and the results into the report. After that, they prepare the datasets and the guidance for the review and submit to the agency to support a new drug application. After we receive it, analysts at FDA will identify datasets and the reports to make sure the integrity of datasets. We need to make sure you have all datasets and the codes for us to review. A lot of times we have to relay the name datasets or create a direction of the structure so to make sure we can repeat your analysis. If we identify problems in this process, we'll issue information requests. How population PK modeling simulation are evaluated? This may be a topic you are interested in. Population PK analysis are checked via standard operating procedure at FDA for data integrity, repeatability, and validation. We do check every dataset and every line of your code after we receive it. We have a range of externally and internally developed tools to use to facilitate this process. Here, this is the flow. We do a population PK review. After receiving the original datasets and the population PK datasets, we check data quality. We in front of in our computer system, we have complicated computer system and a server. Not just check repeat your analysis. A lot of times we conducted the independent analysis with the tool we have. We also conduct additional analysis to check the performance of your model. All of these results eventually will translate into a regulatory decision. Here, we highlight the challenges in conducting population PK analysis. This slide shows the different stages of drug development from preclinical, phase 1, phase 2, phase 3 until filing. All of these stages have different objectives and different target and these challenges. And the population PK and the other quantitative analysis tool will be used across this development process. The challenges in conducting population PK analysis is lack of standard datasets and tools. For as you may understand as a pharmacometrician, the population PK development is an evolving process, and the model will be frequently refined when more clinical data becomes available. Without a standard data set or standards in data sets and the tools, it's very hard to update the data sets, or it will be very time consuming. To address these challenges, we use a strategy similar to ADPPK. This strategy has been used in the last 10 to 15 years in the division to create CDISC compliant data structure for a single or multiple studies for population PK analysis and for disease database or disease models. We have other data sets similar to ADPPK. The advantages of ADPPK is very evident. For example, is analysis ready. The ADPPK datasets may be used to create tables, listings, and the figures. And this output may include time concentration time profile, tables of demographics, or covariates that could be included in analysis and a report. Datasets like PPK is software independent, while this datasets was heavily influenced by Longman. Longman is still the gold standard for conducting population PK standard analysis. We have no endorsement of any specific software or programming. The ADPPK guaranteed the source data can be traced. In the early days of conducting population PK, Longman ID will be different from unique subject ID, and a lot of times we have to issue information request if we cannot trace the original dataset. So you may be wondering, what's the next for the agency? We are very excited about the introduction, of the ADPPK. We had several rounds of internal discussion, and will continue to have discussion on reviewing and the utilization of the ADPPK datasets. We collected feedback and discuss how to update our guidelines using this dataset. We update our model and data format websites regarding preparation and submission of ADaM ADPPK datasets for NDA or BLA submission. The reason to doing this is to ensure transparency for all stakeholders. Your future discussion regarding reporting of standard population PK parameters is being discussed with the ISoP. Here are some take-home message I want to deliver. As mentioned, and the release, we are very excited about the release of this datasets. This is really a major milestone, a major step forward, not only for pharmacometric community, but for regulatory agencies as well. It may change the way we conducted population PK analysis. We believe the implementation of this standard for population PK analysis will improve data quality, facilitate tool development, and accelerate collaboration across pharmacometric community, and enhance community communication with the agency. As always, we encourage early discussion on plans for submission with the agency if you have any questions or uncertainties, and early communication with the agency and ensure smooth submission of population PK. At last, I want to acknowledge many staff members, reviewers, and the leadership involved in the development of this ADPPK. We review involves in the whole process of developing this ADPPK. The office involved in this process had the agencies include the Office of Clinical Pharmacology, Office of Strategic Programming, and the Office of Business Informatics. Specifically, I want to thank our former and current colleagues and leaders, Dr. Jim Lee, Dr. Jeffrey Flory, and Dr. Yanni Wang, the former division director, and currently the Dr. Zhang Liu and the division director, Dr. Hao Zhu, for their support. With this, I can thank you for your attention. With this, I conclude my presentation, and we open the floor for question and discussion. Thank you.
Thank you, Fang. So I'll start by opening the Q&A box. So there's a question: can ADPPK be directly fed into non-MIM or any other analysis system? Yes, it can be. So it has numeric and character variables, but you could always push the character variables to the rightmost side of the data set and use ignore or drop or something like that in your non-MIM control file to drop those variables, and it can be directly used in the non-MIM. And we have another question: how about monoliths accepted by FDA? So Fang, do you want to take that question?
Yes, I want to take that question. The answer is yes. Monoliths is accepted. We don't have a specific endorsement for software, any software. So monoliths and Phoenix is AME is also accepted. But at this time, about 90% of submissions still non-MIM. But we are okay with the monoliths or any other software. We have no specific requirement.
Thank you. Thanks. We have another question about: can you comment if some variables are considered required versus optional in ADPPK? Yes. So if you can see my screen, I just popped up the implementation guide. I didn't go through the whole structure of it, but this is how the implementation guide is, just like any other SDTM ADaM IG. So if you see the core column in here, you will see something called perm or rec, or cont. So required variables are labeled with REQ in the core, and permissible variable, which is optional, is PERM, and some of them are conditional based on the usage. Hope that answers your question. And I'm going to come to the raised hands after I finish the Q&A in the box. So there's question on: although we have been consistently using SDTM datasets for PopPK dataset build, of late some folks ask us to create PPK using ADaM. Does FDA have a preference of using ADaM over SDTM while creating PPK datasets? I know this floor is for ADPPK, but this impacts both PPK and ADPPK dataset build. Is there a preference to use ADLB over LB or ADSL over DM and so on and so forth? This question may be answered by any panelist, and we would like to hear from Mr. Fang as well, as he's in the FDA. Thank you. So, Fang, did you get the question, or should I rephrase it?
Yeah, I see that. So SDTM is the original data set for each single studies, right? ADaM is derived data sets from the SDTM. Actually, all data sets was originally from SDTM, and SDTM this required data sets to submit it there. We don't mind which way you create your data sets. We just want to make sure this is when you create population PK data sets, we just want to make sure this data sets can be traced back to any SDTM. You have subject ID that's included in both SDTM or other ADaM data sets, ADSL or something. So yeah, it's fine. If we can trace the back, if we have subject ID, we ourselves can trace the back to SDTM as well. I hope this answers your question. But when we discuss this ADPPK, we do have people ask, well, if I use ADPPK as a long-man data sets, and if they are different, what shall we do? We say, well, shall we submit both of them? I say, yes. If you have ADPPK and long-man data sets, you can submit both of them. If you use programming for for variable selections there, you should include your programming that derive ADP, your long-man data sets from ADPPK.
So I would like to comment on this question as well. The specifically asking SDTM versus ADaM or ADSL versus DM. So in my experience, ADaM is the most updated one because let's say you pick a baseline value from SDTM versus a baseline value from ADSL. There is a chance they're different, because there's some criteria or some algorithm might change and ADSL might use a different way of deriving baseline values. So that's why even in the implementation guide for ADPPK, we've talked about it and we said immediate predecessor. So if ADaM ADSL is available, we recommend using them because that has the most up-to-date information. But based on when you start your programming for PopPK, if ADaM is not available, you could start with SDTM. But, the recommendation is to compare if, your results are matching with what's reported in CSR. So that's the key. Moving on, at some point, will the FDA require an ADPPK with all PopPK analysis? Fang.
Well, at this time, is recommended, not mandatory. I know this question. So we encourage you to submit to use ADPPK. But if you still use at this time, especially this transition period, will not reject it. But we look forward to receiving more ADPPK so we can accumulate more experience. At this time, we do not have policies, so you have to subgroup ADPPK. Yes.
Okay. And there's a question about, do we need a special data spec and define submitted with ADPPK? You could follow the define.xml that we use for other ADaM submission, or you could start with the regular define.pdf and then transition to define.xml at a later point. Yes, we will provide the URL for the PDF. And some support. And there's a question, do you anticipate proposing an ADaM standard for ER? I mean, personally, I don't know if we want to because ER datasets are very different, for different therapeutic areas with different endpoints. I don't see much value in coming up with a standard that maybe only have limited use because of such variety of the ER datasets, but we can talk about it. What are the timelines for the implementation of ADPPK as required, accepted by the regulatory agencies? Fang?
Yeah. I think there's a question.
You already addressed it.
Yeah. Yes. My answer is yes. 2 months accepted. We got a training on 2 months.
Okay. Is the ADPPK meant to be generated per study or per analysis? Is there an example of ADPPK pooled with studies? Yes. You would start by generating per study and then you pull them all together, just like any other PopPK data set that you have been doing together. Yes, we have examples in the implementation guide. One of them has pooled studies and we can point. So let's go to the raised hands question. So please speak up.
There's a quick question, I think, related also to the FDA submission aspect. So like you said, Neelima, this data set can be used in something like non-MEM, basically as is. But do you expect in some cases we would have the actual data set that we're entering non-MEM to not have to do some of those adjustments you talked about, like in the control stream, and then also have the ADPPK kind of side by side?
So sorry, what was your question?
Yeah. So when you go to submit, do you ever imagine a scenario where you'll submit, as part of your submission, you'll have your non-MEM CSV file that you actually ran in the modeling, and then you'll also have an ADPPK on top of that. It's very similar, but it wasn't actually run in the model. Maybe the columns are different orders and things like that.
Right. Yeah, I mean, Fang's opinion from FDA perspective is they're okay if we submit both, but my personal preference is to stick to one data set to avoid any discrepancy between two files. And we've been doing that for over years and it works fine maintaining one file. But again, people have different preferences and looks like it's okay to submit both.
Yeah, some both. If you have your long-run data sets and ADPPK data sets is a lot different. If you use the programming, SAS programming or R programming to derive your Then for ADPPK, you'd better submit your code as well.
Yes. Okay. That makes sense. Yep.
Yeah, so they can reproduce how you landed there. Yeah,
Yes.
with the code.
Oh, that makes sense. Thanks.
Some people talk about what's the transition period. We do not have a definition of transition period. I think this takes time. From this time to maybe I think at this 1 year is a transition period. As I said, if you have question regarding your submission, we have pre-NDA or pre-BLA meetings there. You can raise the question there, and we'll provide answer.
Okay. Any other questions?
A quick question is, can we have access to the slides?
Yes. ISoP is going to release both the slides and the recording of the webinar to all the participants. And then I'm sure it will share in the link.
A related one, the document you're mentioning, showing right now, you said you may be forwarding this as well. Is that correct?
Yeah. So this document is part of the CDISC website, but like I said, not everyone has an account on CDISC. So unfortunately, you need an account to download this implementation guide. So what I could do is, since this is all of PMX community, I'm going to download a copy and put it on ISoP so they can access it from there. And when it gets updated, we'll make sure to, keep it updated. So that was a very engaging discussion. I'm really excited and energized by all your interest, and I'm looking forward to everyone adopting these standards and, making everyone's life better and streamlining the collaboration across the companies. So looking forward to seeing that. And thank you, Dr. Fang, for sharing your insights from FDA. And thank you, Luke, as well, for sharing your insights on CDISC and SDTM.
Thank you, Neelima.
Yeah, thank you, everyone.
Thank you all.